and go to Dr. Shilpa iYer, who will be presenting on preclinical stem cell models for Leigh syndrome.
Can you hear me and see my screen?
Yes, we can hear you and see your screen.
Okay. Um, so thank you so much for um, uh, providing me this opportunity and apologize for the crash and the technical glitch in the morning. Uh, what I'm really going to talk about is very relevant, I think, after the talks that we've heard about nutrition and right now the current talk on repurposing drugs. So we are working on creating preclinical stem cell models for Leigh syndrome. And without much, description, I do not want to delve too much into it. We've been hearing excellent talks on Leigh's and how heterogeneous it is and how many different genes are impacted in Leigh's disease. So mainly, just going overview is that the neurological the brain is affected, and then you also have the GI issues and cardiac and other abnormalities besides, of course, the genetic mutations. So, when we started this work, we wanted to address directly what the patients, what we heard Anju's early talk was how do you address the unmet need of widespread mitochondrial or energy dysfunction and where in various cell types and how it was impacting the functioning of different organs. And we felt, as we all heard, that there aren't many good clinical, good animal models at that time. So we decided to create customized patient stem cells and their derivatives so that we could, provide the answers that the patients were seeking. And the way we wanted to start was a non-viral reprogramming strategy so that we could create clinical grade patient-specific stem cells. And from there, we could create, differentiate them into different organs and tissues that were impacted in that particular patient. So this is a picture that of how we took 5 different Leigh's. This is an example since it's a Leigh's syndrome network meeting. So I'm giving you example of 5 different pediatric patients with Leigh's disease. And we took skin samples from them and reprogrammed them and converted them into stem cells by 15 days. So you can see that these are fibroblasts on day 0, and by day 15, you can see them, converting into induced pluripotent stem cells. We also checked for different markers for pluripotency, and all 5 of them, BJ is the control, and then we had the 5 lines. I'm sorry I haven't shown you another one, but they were all, positive for the pluripotent OCT4 and SOX2. And I need to say that SBG1, 2, and 3 are complex 5 defects. The SBG4 and 5 are complex, sorry, yeah, a complex 1 defect. That's true. So the first thing that we wanted to analyze is when we saw the iPS cells, we wanted to look at, do they indeed have mitochondrial morphological differences and could we detect them? So we came up with a very simple, it was an undergrad project, so it was a very visually appealing sort of project. And they worked hard and found out that there were multiple different morphologies that the mitochondria exhibited. One was individuals, the other one was they had, branched networks, or networks, and then you had branch networks that were a little bit more complicated. And then we had an elongated network and an elongated branch network. So we kind of designated individuals and networks in the normal category, and then branch networks and elongated networks as undergoing fusion. And we had elongated branch networks as hyperfused. So when if the mitochondria showed a hyperfused morphology, or they were showing more, individuals, we call them fission or, hyperfused, and the other one was typically normal. And now, just summarizing that, what we kind of saw was in those 5 lines that one of the mutations, the one of the patient skin cells exhibited more fission-like morphology, but most of them fell in there were two that were, showing elongated networks, and there was one that was hyperfused with a branched sort of network, elongated and branched network, and one was in a very physiologically normal, like the um, control. So we kind of came up with a hypothesis, or we kind of concluded looking at this, that the shape of the mitochondria was very much dependent on, they were either fragmented or fused in Leigh's syndrome, and they were dependent on the capacity to adapt to stress and the mitochondrial membrane potential. So we measured the way we measured the capacity to adapt to stress was measured mitochondrial spare reserve respiratory capacity. And we saw that if you look at this particular slide, the left shows, basal respiration or mitochondrial oxphos, and the right shows the glycolysis, or as you would say, proton efflux rate. But in the middle, that is basically what you can see, is that all of them are showing a decrease in spare reserve capacity at, although different levels, but you can see that they're all significantly reduced. So this, if I were to, extrapolate this and convert and look at the healthy control fibroblast as 100%, and orange is your spare reserve capacity, blue is your proton efflux rate, or a cellular respiration, and yellow is your mitochondrial. So you can see that different, although Leigh's, they show different mitochondrial capacities, and those that are lower, yellow, lower, and then they kind of adapt to a more glycolytic if the spare capacity is higher. But here you can see that although they adapt to a higher glycolytic phase, but their spare capacity is low. And those lines with lower reserved spare capacity or ability to adapt to stress also had mitochondrial fragmentation. That was, they were either fragmented or they were fused. So the morphology was very much linked with more mitochondrial spare capacity rather than basal respiration or glycolysis. With that, we looked at membrane potential, and we saw that this was a more reliable marker, where we saw a decrease in mitochondrial membrane potential. Some of them were statistically significant, and there was one that was trending to be lower, but was not highly significant. But it overall, we saw a decrease in mitochondrial membrane potential in these stem cell lines. So we started to look at the two main organs that are impacted. They are highly energetic and impacted, at least the one neurons. And we saw that when we use different inhibitors, that is the BMP is it inhibits the other, does not inhibit the ectodermal differentiation into neurons. We saw that with and without these inhibitors, the number of neural rosettes and the capacity of neural rosettes, if you look at it, if you look at E6 and E6 with the inhibitor, you can see that when you inhibit the other two germ layers, the capacity of cells to actually form neurons increases. So it really matters. You can manipulate a little bit using different medium to figure out neural differentiation and early differentiation in Leigh's disease. That's our finding, but we are working on trying to figure out how astrocytes and oligodendrocytes, also the glial components, are impacted. Then this was a very surprise finding, and I kind of was thinking about it from a cardiac. I didn't realize I wasn't expecting cardiac to be impacted, but one of the lines, the one that was hyperfused, did show an increase by the time we differentiated them into cardiomyocytes. By day 30, it showed an increase and almost erratic beating of the heart. We took videos. This is a small video of how we ended up even measuring the heartbeat, which is visible on in a dish. And what we saw, the other one, I was expecting it to be like the control, but apparently by day 30, the one that was classically Lee's also demonstrated a reduced beating. So, which showed us that we do not really see this effect, but if you were to measure it, you can see that the heart cardiomyocytes are impacted in Leig's disease, at least the two of them. Now we are looking at the other three lines to see if they also show the same kind of erratic heart, cardiac differentiation issues. So this is a new thing that we saw in our own lab. And I think we heard a talk just before mine, and we also observed urea cycle disturbances. And this is just one example I'm giving you, because SBG1 is a Lee's fibroblast, and we saw elevated polyamine levels in when we did a metabolomic analysis, as you are hearing that nutrition and mitochondria are intimately linked. And we did see an elevated level of putrescine in all the fibroblasts, as well as not just Lee's, but we also measured in Kearnsey's, in Pearson, in Milas, and we saw that the putrescine was elevated, and there was a decrease in the NAD/NADH ratio. So this was a very surprised, not when we saw that, we were very excited, but at the same time we felt, oh, we should have looked at this more carefully. So this is a new marker, if you might say so, and we hypothesized that elevated putrescine levels causes this polyamine increase due to widespread mitochondrial dysfunction in different mutant types. And we are now looking at differentiating them into neurons, cardiomyocytes, kidney, intestine, and liver cells in Leig's disease. So an overall summary is that we see impaired bioenergetics, altered mitochondrial structure, and defects showing up in early development, because the induced pluripotent cells are markers of early development. The neuronal and cardiac differentiation was impaired, but neuronal could be manipulated by different inhibitors, whereas cardiac did show direct by 30 days, was showing either increase or decrease in the beats frequency. And by selectively targeting, so now if you're looking at drug repurposing or different drugs, so I think where we are heading is we are trying to target the polyamine metabolic pathways, which will lead to better diagnosis and focused drug therapies for primary mitochondrial disorders. And with that, I would like to thank my members of my lab. Some of them, Josh, Ajibola, Phoebe, and Indra Chappa, are the main data that we have shown from all the pictures that I showed for Leig's disease. The others have been involved in other mitochondrial diseases, and we have collaborators from UGA, Wisconsin, and Arkansas, and also Virginia Commonwealth, who are involved in, and Olivier Finn, who have shown us, who have been, working with us to provide a lot of input and cell lines reprogramming and a lot of technology help. And we are very, very grateful to Children's Hospital at Innsbruck and Salzburg. That was the SBG, are the Salzburg lines. And we also have the North American Disease Foundation lines. And we are grateful to Amel Sumit for their, advice and help throughout our project. And our research has been funded by Arkansas Biosciences Institute, NIH, NICHD, and DoD. And with that, I think I would like to thank all of you and take any questions.